Right, welcome to this recording about enzymes, and I'm trying to do a little podcast here, a webcam or whatever, to go along with this so you can see my nice cheery face. It will come and go, you'll be glad to know, so you won't have to see it the whole time. Anyway, we're going to be looking at both the outcomes at National 4 and National 5 in this video. National 5s have already looked at some extra bits that they need to know about enzymes, so some of this video will have a little bit of a repeat of what they've already done, but it also then goes on to cover specific enzymes. So first of all, we're going to have a look at what is an enzyme. Well, an enzyme is known as a biological catalyst. By a catalyst, we mean something that speeds up a chemical reaction. So a biological catalyst is something that speeds up the reactions in living cells. Your cells are full of enzymes. If they weren't there, then the chemical reactions that take place in them would go too slowly for the reactions in order for you to stay alive or for the cell to stay alive. So they're absolutely essential. Other things that you need to know about enzymes, well, these are called properties of enzymes, and what we mean by that are kind of features of them. What are they like and what they do? So they're made of proteins. They're involved in chemical reactions and helping them speed up, but enzymes themselves aren't changed by the chemical reaction at all. They just help to speed up the reaction. They themselves stay the same. Enzymes are known as specific. That means that only one particular enzyme will work with one particular substrate. And I'll just go over some of the terminology that you need to know for that. What do we mean by a substrate? Well, a substrate is the thing that the enzyme works on, the substance that the enzyme works on. And the example that I've got here is an enzyme that's called amylase. And the substrate for amylase is starch. Now, I said on the slide before that enzymes are specific. That means that only one particular enzyme will work with one particular substrate. So if we have a little look at amylase, amylase therefore is specific to starch. It only is involved in the breakdown of starch. So that's what I mean by specific. The other term that you need to think about is the product. So the substrate is the thing that the enzyme works on or changes. The product is the thing that is made, the substance that's made by the reaction that's taken place. And in the example of this amylase and starch up here, the breakdown product from starch, and amylase helps to speed that reaction up. The breakdown product is maltose, which is a smaller sugar. Because starch is a very, very large carbohydrate, maltose is a smaller one. So we're going to go on and have a look at some specific enzymes. And we're going to have a look at this one here. This is an enzyme that's called catalase. Now, catalase is found in animal and plant cells. It's found in every living cell. It's needed to speed up the breakdown of something called hydrogen peroxide. Now, hydrogen peroxide is produced in respiration in cells, and it's actually toxic to the cell if it's there in high amounts. So it has to be broken down. And it actually breaks down just very, very slowly itself, just to oxygen and to water. But the problem is that it does that very slowly. And so inside a cell there's got to be some catalase, the enzyme catalase. And it helps to speed up that breakdown. So what happens is the actual substrate for this is hydrogen peroxide. The enzyme is catalase. It breaks down the hydrogen peroxide to the products, which are oxygen and water. And a good way that I always say to help to remember this reaction is to call it HP-cow. In other words, hydrogen peroxide, that's the letters in the front, is broken down by catalase to oxygen and water. Now you need to know this enzyme, and you need to know the substrate, and you need to know the products. that are formed from it. And we'll have a look in class at some experiments that go along with this. The next enzyme that you need to know about is the enzyme amylase. And amylase it's actually found in your saliva in your mouth. It's also produced by your pancreas, and it's a breakdown enzyme as well. And it's involved in breaking down starch to maltose. Remember, we talked about that before, that amylase was specific to starch. So here's the word equation for it, and a good way to help remind you of this is to look at the word equation. Starch is broken down by amylase to maltose, and we can use the term SAM for that, for starch, amylase and maltose. Now, one other enzyme you need to know about is this enzyme here, which I'm hoping is going to come up in the next slide. It's just taking a long time to appear. Not quite sure what it is I'm doing. Oh, here it is here. It's called potato phosphorylase. Now, potato phosphorylase is just a little bit different than the amylase and the catalase. And the reason for that is because it is a synthesis enzyme. In other words, it builds up products. It doesn't break them. It doesn't break things down. It's involved in building up a molecule. And what it does is it takes glucose 1-phosphate molecules and it actually builds them up into starch. And potato phosphorylase, because it is responsible, this enzyme here is responsible for making the starch that potatoes are made up of. And you can actually test for the formation of that starch using an iodine solution. And we're just going to have a wee look at a wee experiment that shows this. But here is the word equation for this. Glucose 1-phosphate broken down by phosphorylase to starch. Sorry, I said broken down. It's actually built up. My mistake. Spot the deliberate mistake. So glucose 1-phosphate built up by phosphorylase to starch. And I usually call that GIPS for glucose 1-phosphate, phosphorylase and starch. Now, I mentioned an experiment that can be done for this, and I'm going to show the setup for it. We'll try and get this experiment done in class. And I'm hoping it's going to move on to the next slide. It's having a lot of bother getting these slides to move over. So here is the potato phosphorylase experiment. And what happens is a dimple tray is set up. And that dimple tray is marked with different times, 0 minutes, 10 minutes, 20 minutes, 30 minutes. And what happens is, in this first row that is here, glucose 1-phosphate and phosphorylase is put in to this row. So phosphorylase. So glucose 1-phosphate and phosphorylase are put into every single one of these dimple trays. And then in the next row, glucose 1-phosphate and boiled phosphorylase is put into this row. Now, a boiled enzyme won't work, and there's reasons for that that we look at later on. And then this last one here, phosphorylase and water, is put into this row. And these times are here because what happens is that at 0 minutes, each one of these, in this row here, each one of these dimples here in the dimple tray gets tested for the presence of starch. So what we're looking for is starch formed. Now, that is done at 0 minutes, and then 10 minutes. Each one of these is tested for the presence of starch. And then at 20 minutes, each one of these is tested for the presence of starch, and then 30 minutes, each one of these. And at the end of the experiment, then you will look to see whereabouts starch has been formed. Now, you actually use potato for the glucose 1-phosphate, for the phosphorylase enzyme. So you actually take some potato and you mush it up and then you filter it to get rid of any starch grains, because you don't want to spoil your experiment by having starch there first of all. And then you add the enzyme phosphorylase. So in this row here, because you've got glucose 1-phosphate and you've got the enzyme, you would expect glucose 1-phosphate to be built up to starch in these trays here. And this one here, you would test it with iodine, and the iodine would turn blue-black as the starch is formed. This one here, the next one down, has got boiled phosphorylase. Now I said the enzyme wasn't working when it's boiled, so you wouldn't expect to see iodine turning to a blue-black in this row here. And in the last one, you've not got the glucose 1-phosphate, so again, you wouldn't expect to see the iodine turning a blue-black colour in this tray either, because there should be no starch formed. So let's have a little look at a dimple tray with the results of this experiment on the next diagram that's here. So you'll see that here on this row here that had the glucose 1-phosphate and the phosphorylase, that as you move along here, as the time gets further along, you'll see that here the iodine is turned a blue-black colour. That indicates the presence of starch. It's starting to form in this one here as well. On the other dimple trays that are there, that's the one with the boiled enzyme, and this one here with no glucose 1-phosphate, but just phosphorylase, you see that there is no starch that's formed in either of these two rows. These are really acting as controls, because what they're doing is they're showing that it is the phosphorylase enzyme that is building up the glucose 1-phosphate to form starch. And it's quite a common experiment to see in exam questions and things, this, so it's good to have some notes on it, and you can always refer back to this video. Now we're going to have a look in a wee bit more detail about the term specific. So we're going to have a look in a wee bit more detail about the term specific. By that we mean that the enzyme only works on one substrate. So for example, amylase will only break down starch, catalase will only break down hydrogen peroxide. So that means that if we try to use amylase instead of catalase to break down hydrogen peroxide, it won't work, because amylase won't break down hydrogen peroxide. Catalase as well, if we try to use that to break down starch, that wouldn't work either. So we're going to move on to the next slide and have a look at an experiment another experiment that you might see that involves using amylase. And you will notice that my webcam doesn't match the words that are here. Looking at this, so here we've got two test tubes. This one here has got amylase and starch in it, and this one here has got water and starch in it. So what happens is these test tubes are left for 30 minutes, and after 30 minutes they are tested, or they're divided into two samples. So like a sample is taken from this test tube. One is put into iodine solution and one is put into Benedict solution. And what you find is the iodine solution in this one here, there's no change showing that starch is absent. So what we're saying here is that the starch that was up here has been broken down, so it no longer shows, as when you add iodine, it doesn't turn a blue-black colour. In this experiment here though, it's using Benedict solution. Now Benedict solution tests for the presence of something called a reducing sugar. That's what maltose is. And Benedict solution is normally a blue colour, but if maltose is present, it will turn, well on here it's got an orange colour. It's actually kind of more like a brick red colour. but it will turn that colour if maltose is present. And you'll see that what happens is, in this here, that maltose appears in this test tube. Now this is showing you that amylase breaks down starch, or amylase is specific for starch. In this test tube here, it's a water and starch one. Now the amylase isn't breaking down the starch because it isn't there. So when these samples are divided and it's tested for the presence of starch, so you add iodine solution to it, you will find that it turns a blue-black colour because the starch hasn't been broken down. And it's the same way when you test with Benedict solution, there's no change in the colour because starch hasn't been broken down to maltose, so the Benedict solution doesn't change colour. So this is showing you that amylase works on starch. This test tube here is acting as a control because you're keeping everything else the same, but there's one variable that you're changing in this. That's the water and the replacing the amylase. And so that's proving to you that it's the amylase that has broken down the starch. So we've talked about cells and the fact that they need enzymes to help to carry out these reactions. Amylase in your saliva that helps to break down the starches in the food that are formed, and the enzyme catalase that's found that helps to break down the hydrogen peroxide that would ultimately kill the cell if it wasn't there. And we also talked about potato phosphorylase that is used by potatoes to make starch or to form starch from glucose when the plant makes glucose from photosynthesis. But there are other uses of enzymes that aren't part of cells, and we make use of enzymes in biological washing powders. Enzymes are added to those, and enzymes can break down proteins that are involved in the stains that are there, and they help to do that, and therefore it means we can wash our clothes at lower temperatures. So that saves energy because we can wash at lower temperatures, saves us money, and it also has the added advantage that it reduces the carbon footprint. In other words, the amount of carbon dioxide that has been used to being produced by energy that has had to be made for the electricity to do your washing. So there's benefits all around, more money in your pocket, and we're saving the planet as well. Enzymes are also involved in cheese making, and there's a specific enzyme called rennin. It's found in a substance called rennet, which is added to the milk, and it helps to clot the milk proteins together and your cheese is made. So that's just a couple of examples. There's lots of other examples where we make use of enzymes in various industrial processes and things like that, but these are the two ones that you really need to know. And that is the end of this video on enzymes.